let you know when it's your turn to start reading.
Oh, okay.
Okay.
Okay. So I think we can start now, exactly at 8 o'clock. So anyway, so my name is Marge. I'm the, between me, Mario Santo Domingo, and Diana Aga, we are hosting this, we call it the introduction and induction of the new members of PAASE. And these are the members that have actually been nominated and elected both in 2018 and 2019. So we decided to recognize them all together. Some of them have been inducted in smaller venues, but we think that this is a good opportunity to showcase everybody and show who are these new members of the academy that have been voted in. So just as a general housekeeping rules, I just wanted to make sure that everybody keeps muted during the introduction and during the presentation, so that we can hear what everybody's saying, and particularly when we introduce the members, so we know who they are. And then we'll give you some time to look at their pictures and their qualifications before we move on, so that we know who these people are. And then at the time that we do the induction, we're going to ask those who are present. There are several of them who are present. We actually have 2 induction events this morning and this evening. We want to ask those who are present to please turn on their video and turn on their audio, so that they can recite with Giselle the oath of the membership. And then after the oath, when I take off the slides, we're going to take a picture of everybody. I guess Richmond can do that, or I can do that. Somebody will do that, so we can have remembrance of all of these. Okay. Okay. So we're really very excited to welcome these new members. As you know, our organization is as good as our members, and they are the ones who define who we are and what the direction we are heading for, which is, as you have seen in the last few weeks, is really full of excitement and hope and a lot of new things to come for PAASE. And so these members that we are going to introduce and induct today have all distinguished themselves in their respective areas of expertise. And that's irrespective of whether they've been in their profession for many years, or if they have just been starting. They have distinguished themselves, and that is why they have been elected as members. And as you will actually see, once we show you their pictures, you will recognize a lot of them, because as they have become members, they have already become active members. They have been chairs of sessions. They have organized the sessions. They presented their research. And I have to say, I don't know about you, all of these has been very exciting to listen to. And I've looked forward to every evening to listen to all of these presentations. So these members have been inducted, have been voted, and they have already shown that they are active members of our organization. So last year, when we were planning the 40th anniversary of PAASE, we were so excited to gather in the Philippines and gather in the US and meet each other and chit chat and talk and share ideas, as we often do when we gather together. But unfortunately, this pandemic upended all our plans. And I have to admit, a few months back, when Giselle brought up the idea that we are going to go virtual, she said, and we will have talks for a whole month. And we were like, what? For a whole month? Aren't we going to get tired? But as I have just shown, and as we have seen, this has been some very exciting presentations that we've had. And in fact, it has brought together a lot of people from all corners of the world. And the pandemic actually made it possible to bring a lot more people who otherwise wouldn't have been able to fly to the Philippines and share their science, not just with those around them, but through everyone through around the world. So my misgivings were proven to be wrong. And I think some people have actually said we should keep doing this. This should be the beginning of many new things that we have found. So the enthusiasm, the participation and passion that everyone has shown, whether they are presenter or participant, has truly surpassed a lot of our expectations. But more importantly, what it showed was that we are the life of this organization, and the vitality of our organization cannot be dampened by things such as a pandemic that we know will hopefully leave us the sooner the better. So what is really inspiring is the fact that a lot of these members that you'll meet are the ones that will keep PAASE moving forward. And it showed us that even though we are apart because we cannot be together, this symposium that we have is really a mechanism that allows us to promote science and technology in the Philippines and around the world by virtue of us being engaged and working together in collaborative means. So with that, as we welcome these new members, I would like to invite you as chair of the membership committee, not just to look at these members, but to also call upon your colleagues to become members of the organization. And it's not just a matter of applying to PAASE, but someone has to nominate them. So nominate the people that you think are worthy of being members or who can contribute a lot, and send the nominations so that the membership committee can look at them and present them to the membership to vote them in as the future members of our organization. Now, because of the enthusiasm, I have sent out the call for nominations sometime back a couple of months ago. Because a lot of people wanted to nominate, they have asked to have the deadline for the nomination to be extended. So instead of the deadline being August 31st, our deadline for submission of new nominations for the full and associate members will be extended until September 15th. Okay, so after this induction, what I'm going to do is to send out the call for nominations again. I have already received quite a few nominations, and I'm told that there are more coming. So please go ahead and fill those up and send them to me. They're all going to be sent to my email address, which will be in all of these nomination forms, just to make sure that they all come to one destination. Because sometimes back, we have some nominations that were quote unquote lost in the ether, in the internet, because we didn't know where they were. They were not really lost. We had to go find them. So this time, just send them all to me, and then the committee can go over and review this. Okay? So watch out for the email. I will resend that with the nomination form. So now, the other thing that I would like to do is to invite you all to attend our business meeting, which will take place on August 13th. So this is important. At this meeting, this will be led by the chair of our executive board, Carlito Librilla. But at this meeting, we want to summarize the initiatives that the board, the executive board of PAASE has initiated and carried out in the past years, what the accomplishments are. The biggest of this is actually getting PAASE, which is a big organization, and forming the research expertise clusters to encourage collaboration, particularly among individuals that have shared research interests. So please come to that meeting so we can tell you what we have done, what we want to do, but more importantly, please attend the meeting so that we can hear your suggestions, that we know you are there and we are here, and it would be great to see what ideas you have as to how to make PAASE a more active, cohesive form and be able to accomplish a lot, not just for our own benefit, but most importantly for the benefit of our country in the Philippines. So that's important. Please be there. We look forward to seeing you. And so at this point, without any further ado, before we go on, I would like to acknowledge Dr. Isadora Yu, Isa, and Hias Yunyo. The slides that you're going to see today, the PowerPoint slides, were put together by both of these young scientists who are also going to be inducted today. You can see they're already doing their part. They actually volunteered to do that. And so I want to thank them for putting this together. It was a big effort to get everybody's pictures and qualifications and everything. Also, I would like to note that these PowerPoint slides that have the members in it will be posted on our webpage by Richmond, our very able PAASE administrator, so that you not only see them there, the people who missed this event will also see who are the new members and how they can begin to form connections and collaborate with them. Okay, so without further ado, I want to introduce Dr. Mariano Santo Domingo. He was the chair of the PAASE membership committee in 2018. That was a time when we had over 100 nominations, and he did a valiant effort of putting those together and getting us to evaluate them. And so Mario is going to present a very short history of PAASE. Okay, Mario?
Thank you, Marge. So I'm going to show you few historical tidbits regarding the origins of PAASE. As many of you know, but I'm sure probably some of you, especially the students, are new to this. PAASE is, or the Philippine American Academy of Science and Engineering, is a non-profit organization composed of scientists and engineers of Philippine descent who established themselves in communities. It was founded in 1980. The aims of PAASE are to promote the advancement of science, engineering, and technology, encourage collaborative work among scientists and engineers of Philippine descent in research and development through scholarly and scientific endeavors, support efforts that advance science and technology, and recognize and honor the achievements of scientists and engineers of Philippine descent. So when did PAASE, when was it founded? It was founded in 1980 by Dr. Severino Cole. Severino Cole then was a professor at Purdue University at West Lafayette in Indiana. And these are the incorporators, according to his recollection of the origins of PAASE. He met this gentleman in a wedding banquet because he was looking for scientists and engineer scientists among the Filipino communities, but all he knew, everyone he knew were medical doctors. But then in 1978, when he attended this wedding, he met these three gentlemen, which he encouraged to form the organization. He actually wrote his recollections of the events in one article that he published. And if you're interested, I'm willing to share this with you. One of the milestones of the organization was its inaugural celebration, or the first meeting and symposium, which was held in Silais Hotel, Manila in 1981. So this was a year after the founding, where they invited Philippine government officials, like then the Secretary of National Science Development Board, and then President of National Academy of Science and Technology. And the first set of officers were Dr. Ko. The first vice president was Dr. Jose V. Cruz, and the founding secretary and treasurer was Dr. Linda Gutierrez Bartolome. And these are the founding members, 21, who mostly, if you will be looking at where they come from, are mostly from the Midwest. And that's understandable because Dr. Ko was a professor at Purdue. And so about 60% of them were either faculty members in Midwest universities or alumni or graduate students then. And these are the milestones that I collected from the very limited references or sources of information. I've mentioned already the incorporation in 1980, the inaugural meeting in 1981. Then Dr. Ko transferred to Maryland at UMBC in 1983, where his network grew because of the short distance between Baltimore and DC, where there are federal agencies like the NIH, NSF, and of course the Department of Agriculture and other federal agencies. One other milestone is the establishment of the Founders Lectureship Awards in 2000, and then it was renamed as the Severino and Paz Co Lectureship Award in Science in 2004 upon the death of Dr. Ko, and then they added the Lectureship in Engineering Awards in 2008. Between 2006 and 2007, the PAASE Philippines was founded, and the first Philippine-based president was elected, and it's Dr. Alvin Colaba. This year, we've had like several constitutional amendments, like the changing of the term of the president from 1 year to 2 years. And then, of course, our still ongoing 40th PAASE anniversary and annual PAASE meeting and symposium, which is still ongoing. These are the lists of the lectureship awardees between 2000 and 2020. The most recent awardees are Dr. Danilo Tagle for science and Dr. Just Santos for engineering. This is very important, and thank you very much, Dr. Gisela Concepcion, for reminding me of this very important milestone. In 2006. There was a PAASE campaign in Philippine Congress, the Senate and executive branch, resulting to the establishment of the National Complex in UP Diliman and more funding in DOST for MS, PhD scholarships, and R&D projects. As a result of this campaign also, many PAASE members started coming over under the DOST Balik Scientist short-term program to serve as consultants or advisors. Now, I also, I'm showing here the list of past presidents. There have been 37 of them, men and women. The first woman president was Dr. Victoria C. Guerrero Abelera in 1998. And the first Philippine-based president, as I've mentioned already, was Dr. Alvin Kulaba. And then there is this tradition in PAASE that the president alternates between the US and the Philippines. These are just some photos of the APAMS that was last done at in Manila. And we miss this type of event. Of course, in the future, we look forward to where we can actually meet each other physically and have the APAMS either here or in the Philippines. And these are our current PAASE officers, our President, Dr. Gisela Padilla Concepcion, Vice President Edsel Peña, Secretary Lourdes Harold, and Treasurer Ana Cristina Sarquina, and our 9 Board of Directors. Thank you so much.
Thank you very much, Mario. It's always good to hear about the history of PAASE, particularly for those who are just starting out to be part of the group and knowing where the history is so that we can define where we are going to go. So I'm going to introduce the next part of this program, which is Dr. Diana Aga. She was actually our co-lecturer awardee in science last year. And Diana was also chair of the membership committee in 2019. She will describe to you what is the process for becoming a PAASE full or associate member. Diana?
Yes, good evening, everyone. It's really nice to be a part of this induction tonight. So I've been actually a member of the PAASE for probably about 10 years now, and it's been really a nice organization. So I've been quite active with it in the past few years. So I just will talk to you briefly about how you become an associate member and how you become a full member. So let's discuss first the associate membership. So to be an associate member, you should be a little bit earlier in your career. So typically you should be within no more than 10 years in your terminal degree. And this means all the associate members that are nominated have doctorate degree as a PhD or doctorals or equivalent in science and engineering. So to become a PAASE associate member, it is a 3-step process. First, you have to be nominated by an active member of the PAASE. And then your pre-election will be vetted by a membership committee. So we have a committee that evaluates all the nominations. And then once the membership committee recommends your associate membership, all the members will vote. And to be officially become a full associate member, there should be about 66% of the PAASE members to vote yes. So this is a highly vetted membership. And there is, to be a active PAASE or associate member, you should have demonstrated the ability to do scientific research and show promise for achieving distinction in some fields of science and engineering. So how do you become full member? You should be an associate member for at least 5 years to be recommended to full membership. And also to become a full member from being associate member, you need to be able to present in one of the meetings, either online or in person, before you can qualify to become a full member. Okay, and you should be nominated again for a full membership, and and also there's a bylaw that you should not be more than 10 years as an associate member, because if you fail to be nominated to be a full member, that means we interpret it as lack of interest to pursue further PAASE membership. So you need to be an active member, even as an associate member, to become a full member. Okay. So now, how do you become a full member? So the applicants for or nominations for full membership, you must have a doctorate degree or terminal degree equivalent to a PhD in science and engineering. And you should be nominated by an active member of the PAASE. And to become a full member is a four-step process. So first you have to be nominated, and then you have to be pre-elected by the vetting membership committee. So your application or nomination will again be looked at and evaluated by a membership committee. And then it will be presented to the full members and be voted by at least 60% of the PAASE member. And finally, you should be presenting at the APAMS to become a full member. So that's a requirement. But I'm really happy to see that a lot of the current associate members have already done their job in presenting. So I think it's a very easy requirement to fill. And so according to the PAASE bylaw, members shall be restricted to scientists and engineers who have distinguished themselves in their respective fields through research publications or technical achievements. So these publications have to be peer-reviewed, journal articles, patents, books, technical reports, and others. I think that's it. A lot of them are just things, but those are the most important things. So hopefully the members, the associate members that will be inducted today will become always active, and we will see you within 5 years to become full members. That's it.
Okay, thank you, Diana. And so Diana has fully explained the process for that. And so the first step will be the nomination process, of course. So if you know of anyone who is willing and very interested and has a passion for their research, we welcome them. And please send their nominations to us so that we can review them in the membership committee and then recommend them for voting. And as Diana had said, many of the members that you will introduce today have already fulfilled the requirement of presenting a talk at one of the APAMS meeting. It was one of those requirements that we added a couple of years ago and caught a lot of people by surprise, but we think that this is a way that we can actually engage you. I remember being elected as a member and I didn't know anybody, and on the first meeting that I went to was in NC State, and it was really a revelation to meet and really exciting to meet a lot of our fellow citizens from the Philippines and those who are of Philippine descent doing such outstanding science. Okay, so now we come to the point where we are going to introduce the new full members. So I will return the chair over to Mario, who will begin the slide presentation.
Hello again. So this is Mario. I will be reading the names, showing the slides for each member for about like 10 seconds to allow our audience to read what's on the slides. So let me start. The members can say hello or hi if you so wish, so that you'll be recognized.
If I may say something, the first few names will be the ones who are actually here present. So yeah, you can say hi if you want to.
Dr. Inmaculada B. Aban, Professor, University of Alabama at Birmingham.
Hi.
Dr. Maria Carmen Ablan Lagman, Professor, De La Salle University, Manila.
Good morning. Good morning.
Good morning. Dr. Menandro N. Akda, Professor, University of the Philippines, Los Baños. Dr. Henry A. Adorna, Professor, University of the Philippines, Diliman.
Good day.
Hello.
Dr. Marcus A. Agustin, Professor, Southern Illinois University.
Good evening and good morning.
Marcus.
Dr. Evangeline C. Amor, Professor, University of the Philippines, Diliman. Dr. Rowena Bacongis, Professor, University of the Philippines, Los Baños.
Hello.
Hello po.
Hi.
Congratulations. Dr. Erniel B. Barrios, Professor, University of the Philippines, Diliman.
Good morning, good evening.
Okay, Arniel.
Dr. Emil P. Bolongaita, Distinguished Service Professor of Public Policy and Management, Carnegie Mellon University, Australia.
Good morning, good evening, everyone. Glad to be here.
Emil is a president of Carnegie Mellon University, Australia, if I may add.
Dr. Rinley Butch Cervera, Associate Professor, University of the Philippines, Diliman. Dr. Xiongfeng Anthony Chu, Professor, De La Salle University, Manila. Dr. Edward Vincent S. Faustino, Associate Professor of Pediatrics, Yale School of Medicine.
Hey, everyone.
Dr. Joel P. Ilaw, Associate Professor, De La Salle University, Manila. Dr. Jose Esagani B. Hanairo, Associate Professor, De La Salle University, Manila. Dr. Jose Florencio La Peña Jr., Professor, University of the Philippines, Manila.
Joey, one of our frontliners in PGH.
Morning, good evening.
Congratulations.
Hello.
Morning.
Dr. Ireneo L. Lit Jr., Professor, University of the Philippines, Los Baños.
Hello, good morning, everyone.
Hello.
Morning
Good
po.
morning.
Morning.
That's Joey.
Congratulations. Dr. Joey Ocon, Associate Professor, University of the Philippines, Diliman.
Magandang araw po sa ating lahat.
Congrats, Joey. TUAS,
Morning,
Joey.
Yeah.
Congratulations.
Morning.
TUAS awardee in NAST.
Dr. Juan M. Pulhin, Professor, University of the Philippines, Los Baños.
Congrats, John.
Welcome, John.
Dr. Ana Cristina Sarquinia, Staff Scientist, National Cancer Institute, National Institutes of Health.
Greetings from Maryland.
Hey, Anna.
And
Congratulations.
And our treasurer.
Dr. Michael Velarde, Professor, University of the Philippines, Diliman.
Congrats, Michael.
biology.
And,
Dr. Al Ray C. Villagracia, Professor, De La Salle University, Manila.
Good morning and good evening.
Hi,
Hello.
Alray.
Hi.
Congratulations. Dr. Krista Danielle S. Yu, Associate Professor, De La Salle University, Manila.
Krista.
Good morning.
Krista.
Hello.
Good morning.
Dr. Percival F. Almoro, Professor, University of the Philippines, Diliman. Dr. Victor Amoroso, Professor, Central Mindanao University.
Good morning.
Good morning,
Hello.
sir.
Hi, Vic.
Morning.
Hi, Victor. Morning.
Hi.
Morning, Vic.
Dr. Pia D. Bagamasbat, Associate Professor, University of the Philippines, Diliman.
Congrats, Pia. Pia,
Dr.
frontline.
Nora P. Caberoy, Lindsay Assistant Professor of Life Sciences, University of Nevada. Dr. Drexel H. Camacho, Professor in Chemistry, De La Salle University, Manila. Dr. Charlotte M. Chong, Research Professor, University of the Philippines, Manila. Dr. Edna Estefania A. Co, Professor, University of the Philippines, Diliman.
Miss Edna.
Dr. Gladys Completo, Professor, University of the Philippines, Los Baños. Dr. Rex Victor Cruz, Professor, University of the Philippines, Los Baños. Dr. Eva Maria Cutionco de la Paz, Professor, Pediatrics and Genetics, University of the Philippines, Manila.
Eva.
Dr. Carlos Primo C. David, Professor, University of the Philippines, Diliman. Dr. Lilian A. de las Leagas, Commissioner, Commission on Higher Education. Dr. Alnar L. Detalia, Professor, Central Mindanao University.
Good morning, good morning.
Hi, good morning po.
Morning.
Congratulations. Dr. Elmer S. Estacio, Professor, University of the Philippines, Diliman.
Gandang umaga po. Gandang gabi.
Elevator. Morning, Elmer.
Dr. Ian Kendrick C. Fontanilla, Professor, University of the Philippines, Diliman. Dr. Remil L. Galay, Assistant Professor, University of the Philippines, Los Baños. Dr. Rosalie A. Hall, Professor, University of the Philippines, Visayas. Diana.
Okay. Dr. Desiree Hautia, Professor, University of the Philippines, Los Baños.
That's this.
Dr. Nathaniel P. Hermosa II, Professor, University of the Philippines, Diliman. Dr. Christine C. Hernandez, Professor, Director of the Institute of Chemistry, University of the Philippines, Diliman. Dr. Marie Lapitan, Research Professor, University of the Philippines, Manila. Dr. May Lim, Professor, University of the Philippines, Diliman. Dr. Priscilla Macan Santos, Professor, University of the Philippines, Baguio. Dr. Pablito Magdalita, Professor, University of the Philippines, Los Baños. Dr. Armand Salvador Mijares, Associate Professor, University of the Philippines, Diliman. Dr. Christopher Monterola, Professor, Asian Institute of Management. Dr. Clito Nanyola Jr., Associate Professor, University of the Philippines, Mindanao. Dr. Prospero Naval Jr., Professor, University of the Philippines, Diliman. Dr. Ricky Nelias, Associate Professor, University of the Philippines, Diliman. Dr. Perry Ong, Professor, University of the Philippines, Diliman. Dr. Isabelita Pabuayon, Professor and Dean, University of the Philippines, Los Baños. Dr. Carmencita Padilla, Professor, University of the Philippines, Manila. Dr. Brian Pajarito, Associate Professor, University of the Philippines, Diliman. Dr. Alisa M. Pelio Alampay, Professor, University of the Philippines, Diliman. Dr. Michael Y. Roleda, Professor, University of the Philippines, Diliman.
Morning and good evening.
Hello.
Good evening. Hello.
Hi, Mikey. Thanks for joining.
Thank you.
Dr. Jacqueline Romero, Senior Lecturer, University of Queensland, Australia. Dr. Arnel Salvador, Professor, University of the Philippines, Diliman. Dr. Annalyn Salvador Amores, Associate Professor, University of the Philippines, Baguio. Dr. Edsel Maurice Salvana, Director, University of the Philippines, Manila. Dr. Armando Sumintak, Professor, University of the Philippines, Diliman. Dr. Maricor Soriano, Professor, University of the Philippines, Diliman. Dr. Rodelio F. Subade, Professor and UP Scientist, University of the Philippines, Visayas. Dr. Sinaida Somalde, Adjunct Professor, University of the Philippines, Los Baños. Dr. Giovanni Tapang, Professor, University of the Philippines, Diliman. Dr. Franco Teves, Professor, Mindanao State University, Iligan, Institute of Technology. Dr. Francisca Tolete Belchek, Professor of Surgery and Pediatrics, SUNY Downstate Medical Center, Brooklyn. Dr. Aristotle Obando, Professor, De La Salle University, Manila. Dr. Magdaleno Vasquez Jr., Assistant Professor, University of the Philippines, Diliman. Dr. Eric Bejarano, Assistant Professor, University of South Carolina. Dr. Edward Wang, Chair, University of the Philippines, Manila. Hi. Dr. Isidora Yu, Professor, University of the Philippines, Diliman. Dr. Mendel Manuel Zulueta, Associate Professor, University of the Philippines, Diliman.
Okay.
Thank you.
Yeah, thank you, Diana and Mario. And so now I'm going to read the names, introduce to you the associate members 2018-19. So let me share the screen. Okay, so these are the associate members that have been nominated and accepted and voted in. So first we have Dr. Erwin Abukayan, postdoctoral research fellow, University of Oklahoma.
Hello po. Good morning.
Hello, good morning.
Hey, Marge, do you want
March,
to make it into a
full
full screen? Yeah.
March.
Oh, hang on. It's on my other screen. Wait.
Okay. Thank you.
Let me just share again. Okay, let me restart that and share that one. Okay, is that good? All right, so next we have okay, now I can't move. Okay, technical difficulty. I gotta get out of this.
Should be in the bottom. There's a
Yeah.
bottom. Yeah.
Hang on. Okay, okay, here we go. All All right, so I have to put it on my other screen, sorry. Screen 2, share. Okay. Can you see this?
No, it's not sharing.
Oh,
Ma'am, is it
sorry.
okay if I share the PowerPoint?
Huh?
Is it okay if I do the sharing of the PowerPoint?
Okay, why don't you do that? Because it's going back and forth between my two screens and I can't seem to get it out.
Can you see the screen po?
Okay, thank you, Richmond.
Okay po.
All right. So we have Dr. Erwin Abukayan, postdoctoral research fellow, University of Oklahoma. Next, Dr. Rodel Barrientos, postdoctoral research fellow, Henry M. Jackson Foundation for the Advancement of Military Medicine.
Hello po, everyone.
Hello. Next, we have Dr. Amy Sherry Barrian, professor, University of the Philippines, Los Baños.
Hi, hello po.
Hello.
Congrats, Amy.
Thank you po.
Welcome.
Dr. Agham Cuevas, associate professor, University of the Philippines, Los Baños.
Hello, good morning and good evening.
Congrats,
Good
Agham.
morning.
Dr. Ronald Del Castillo, associate professor, University of the Philippines, Manila. Dr. Leslie Michelle Dalmasio, professor, University of the Philippines, Manila.
Hello, everyone. Good morning.
Hello.
Good evening.
Good morning.
Welcome, Leslie.
Dr. Lawrence L. Delina, associate professor, the Hong Kong University of Science and Technology.
Good morning, good evening po sa lahat.
Hello, Lawrence.
Lawrence.
Hi, ma'am.
Dr. Angeline Lau, associate professor, De La Salle University, Manila.
Hello, everyone.
Congrats,
Hello, Angeline.
Angeline. Dr. Julius L. Liano Jr., Chief Science Research Specialist, DOST Philippines. Hello.
Good morning po.
Congrats.
Good morning.
Congratulations.
Congratulations.
Salamat po. Salamat po.
Welcome to PAASE.
Salamat po. Thank you po.
Dr. Alan Abraham Padama, Associate Professor, University of the Philippines, Los Baños. Dr. Jomar Rabahante, Associate Professor, University of the Philippines, Los Baños.
Congrats, Jomar and Alan.
Dr. Maha Leia Ravago, Associate Professor, Ateneo de Manila University.
Good morning.
Good morning.
And good evening.
Welcome.
Congratulations.
Thank you.
Okay, next. Dr. Richard Abendan, Chief of Party, USAID STRIDE Program.
Good job, Richard.
Congrats, Richard.
Dr. Joel Adawe, Associate Professor, University of the Philippines, Baguio. Dr. May Joanne Aguila, Associate Professor, University of the Philippines, Los Baños. Dr. Jericho Bacani, Associate Professor, University of the Philippines, Baguio. Dr. Emmanuel Baha, Institute of Clinical Epidemiology, National Institutes of Health, University of the Philippines, Manila. Mary Donabel Balela, Associate Professor, University of the Philippines, Diliman. Dr. Lawrence Bello, Associate Professor, De La Salle University, Manila. Dr. Juancho Colera, Associate Professor, University of the Philippines, Baguio. Dr. Marlon Conato, Professor, University of the Philippines, Diliman. Dr. Sara Daway Ducanes, Assistant Professor, University of the Philippines, Diliman. Dr. Cedric Angelo Festin, Professor, University of the Philippines, Diliman. Dr. Fiorilio H. Galivo, Visiting Associate, U.S. Food and Drug Administration. Dr. Marie Heffern, Assistant Professor, University of California, Davis. Dr. Francisco Hiralde III, Professor, University of the Philippines, Manila. Dr. Carl Robert Handok, Assistant Professor, University of the Philippines, Diliman. Dr. Caroline Marie Haraula, Assistant Professor, University of the Philippines, Diliman. Dr. Hias Junio, Associate Professor, University of the Philippines, Diliman. Dr. Erica Phil Legara, Associate Professor, Asian Institute of Management. Dr. Maria Celia Malay, Associate Professor, University of the Philippines, Visayas. Dr. May Ann E. Mata, Associate Professor, University of the Philippines, Mindanao. Dr. Candy Mercado, Associate Professor, University of the Philippines, Diliman. Dr. Sharon Dunal, Assistant Professor, University of the Philippines, Visayas. Dr. Manisa Paderes, Assistant Professor, University of the Philippines, Diliman. Dr. Marjorie Paharon, Assistant Professor, University of the Philippines, Diliman. Dr. Susan Pancho Festin, Professor, University of the Philippines, Diliman. Dr. Francis Norman Paraan, Associate Professor, University of the Philippines, Diliman. Dr. Irene B. Rodriguez, Associate Professor, University of the Philippines, Diliman.
Good morning.
Oh, hi, how are you?
Morning.
Welcome.
Good morning.
Congratulations. Dr. Sheila May Santander de Leon, Assistant Professor, University of the Philippines, Visayas. Dr. Gerald Tubay, Assistant Professor, University of the Philippines, Las Bañas. Dr. Benjamin Vallejo Jr., Professor, University of the Philippines, Diliman. And so that's the last. Okay, so now we ask the inductees who are present to activate their video and unmute themselves so that they can proceed with the induction with to be led by our president, Dr. Gisela Concepcion. Activate their video and unmute themselves so that they can proceed with the induction.
Need to finish after that section, just cutting up on this. We've lost you, Brian. Can you hear me there? Yeah, sorry.
Hey, Angelo.
Estelle, I have the oath.
We will thank Kabir for his presentation and very well done.
Should we start now?
Hi, everybody.
Oh, congratulations, everyone, and welcome to PAASE.
Oh, can you please take us to the side? Yeah. Thank you, Graham. Thank you.
So, good morning again, everyone. Congratulations.
Can you take a picture of, because I cannot operate it if I raise my hand.
You will take a picture after the oath. Can you stay on so we can take a picture? Giselle, can you see the oath on the screen?
Yes, I'll start now. So
Okay.
this formalizes your entry and commitment to PAASE. And so could you please raise your right hand and after me?
How about that?
We'll start now. I do solemnly pledge to uphold the
objectives Philippine American Academy of Science
Engineering as a full member
and
best of my
pledge
ability
to work to the best of my abilities in a collaborative,
around the world.
members in the Philippines and around the world,
Engineering and
science,
technology,
engineering,
scientists, in disseminating development,
scholarly and
recognize and honor the achievements of scientists,
recognize and honor the achievements of engineers,
science and
and most important, increase the development of science and technology in the Philippines, and the training of scientists
science and
who will shape the future of science and technology in the Philippines.
me God.
Training of scientists.
So help me God.
The future of science and technology in the Philippines. So help me God. So help me God.
So help me God.
Oh, help me God.
Scientists
The development of science and technology in the Philippines, and the training of scientists who will shape the future of science and technology in the Philippines. So help me God.
That's okay.
All right. So thank you all for joining us
That's
today.
okay. Thank you.
Thank
And
you.
Marge,
Okay, so please stay on so I can take your picture. Okay, ready? Say, I'm on page 1. Say cheese.
Cheese.
Okay, good. All right. I think I have to go to page 2 now. There are actually 4 pages. So, all right, page 2. Say cheese. Hello.
Cheese.
Okay. Then I am going to page 3. And then going back to page 4. Lots of people here. Okay, so I think I have everybody. Okay, one more time. So they stay on, keep smiling. So we can post all your beautiful faces.
and I
So
would
one more.
encourage Marge. I would encourage anyone who members to speak. We wish to say something, but I just feel that we're so interconnected this week. So it's yeah, milestone for us. Anyone would like
Okay,
to
so that ends our induction, and we welcome you to the PAASE membership. And so we will mail each of you your certificate of membership to PAASE, signed by Dr. Giselle and the committee chair that you was the chair at the time that you were elected. So we will send that. And then also, please be sure to attend the members of the business meeting on the 13th.
tomorrow.
And right after this induction, we have the rapid fire. So be sure to watch this scientists present their work. Okay? Do we have any final words from Giselle?
Well, Marge, I think that since we have a few minutes before the start of the rapid competition of our poster finalists, we would open the floor for anyone to say something you'd like to say for PAASE, how to make us better as an organization. Just raise your hand or, yeah, be good.
I just wanted to say good morning and good evening to everyone. I was muted a while ago, and it's an honor to be part of the group. Thank you.
Welcome. Welcome.
Honor to have you, Edward.
Hi.
Honor to have you,
Hi,
Ellen.
ma'am.
Welcome, everyone.
Good morning. This is Emil Bolongaita from Adelaide, South Australia. Thank you for this honor, and I'm delighted to join you all in PAASE, and I'd like to express my appreciation to Giselle for her amazing work and for inviting me to be part of this distinguished organization. thank you all. I hope we can collaborate in more ways than one going forward. Thank you.
To Emil.
Thank you.
Giselle, I think Sir Al is raising his hand.
Yes, please. Who is it?
Sir Al, Sir Rafika.
Al Saraf Al, our former president.
Is he a new member, Al Zirafika?
Hello, guys. Just wanted to welcome everyone to PAASE. Again, I just wanted to highlight to everyone that we are one big family of scientists. So use the network to your advantage. Okay? Don't hesitate to contact us, ask for help, ask for people we know so we can help you. So don't be shy. And as you know, Giselle and I have never been shy asking you to do things. So do the same thing to us. Thank you.
And tomorrow we'll talk about how we will reorganize PAASE into flexible groups. And we'll try to find ways to get some funding to our groups, okay, for the operation of the clusters. And also we'll try to get a system going for systematic funding of your proposals. At the start, they will be small projects, but we're working hard to get some substantial funding through PAASE. So stay with us, and hope you're patient, and we get to see a major impact of PAASE on development in our country. And as you will note, we really strive to get many more important, eminent, distinguished social scientists in our organization, because they're critical to all our endeavors, which we know should be interdisciplinary, and science, engineering, and technology can best be implemented and communicated to communities by our experts in the social sciences. I think it's very, very clear to many of us now, especially during this pandemic.
One of the things that we have done was to actually develop this, what we call as the research expertise clusters or research expertise communities, which is really becoming very effective in bringing people that have related interests together. So I encourage all of you, we're going to post the updated member list. It'll be posted by Richmond on the webpage. If you can look over your information, because we don't have all of the information with respect to your interest, if you look at that and just send me any updates to your information, your email, what research clusters you belong to, what research groups, that would be very great, because it will help to facilitate group together and then communicate not as a whole, but in little groups that can work together in a much faster, not a clumbering big group, and move forward. So please update your information. Go check the website. Send it to me, Marge Pena, the membership chair for this year, and so that we can update the information. It'll be easier to put the group together with your input. Okay, so that's one thing that will go live on the PAASE webpage, and then also these PowerPoints will be on the webpage. Okay, we'll know where to find you. Okay, so I guess with that, we want to thank you for coming. Those who decided to come out, wake up early or stay up late, wherever you are in this part of the world. Thank you for joining this induction. We have actually another one later on at 8 o'clock, which is morning in the here and in the or in the US, and the evening in the Philippines, to accommodate those that were not able to show up. So if you want to show up again and say hi, you're welcome to do so. But there's also going to be a data science session going on at the same time. So there will be very interesting things going on. So you can go back and forth between the pages. So thank you again for coming. Welcome to PAASE. And we hope to see you again in all the future meetings, okay? And hopefully when the pandemic's over, we'll see each other face to face, okay? Thank you, guys.
Thank you,
Good job,
Maria.
Salamat.
Thank you,
Bye.
Giselle.
Thank you.
Bye-bye.
Congratulations.
Salamat.
Salamat.
And stay on for the
Salamat.
rapid fire. That will be very
Thank you very
interesting.
much.
Thank you
That's very
po.
interesting.
Congratulations and
Those
welcome.
who want to stay for the rapid
Congratulations,
fire
everyone.
Salamat. Salamat.
Good job.
Salamat po.
Thank
Good
you.
job, Maria.
Bye-bye.
Thank you so
Thank
much, Mark,
you.
Mario, Diana.
Hi, everyone.
Ma'am
I see you.
Giselle,
Bye.
salamat.
Yeah.
Maraming salamat din. Thank you.
Bye,
Okay.
everyone.
Hoping for your active participation.
Yes,
Yes,
ma'am.
we hope to
Let
see
your
all of you.
voices be heard.
Yeah.
Good job, everybody.
Yeah, we
Is Wendell.
Google groups for the different clusters so that it's not huge, so that you can pick up more in your cluster. Yes.
Yeah.
Marge,
Organize it
Marge,
so that
Yeah.
the leaders will
update the ano ha. Si Dr. Perry Ong already passed away. So just update our file.
I think I updated that just recently, but I'll make sure again.
Yeah, but
So I
that's
sent
good. That's-
Richmond the new list, so he should post that soon.
I think those who designed the posters like really decided to include his
That's
slide
okay.
on there.
Yeah, maybe just
So,
an asterisk to say that.
that's right. Just
Posthumous.
Yes.
Oh,
The
okay.
posthumous. The
Just so
posthumous.
good to see Perry.
Okay, I will do that in the evening session.
Just
Yes.
indicate
Okay.
the,
Yeah,
That's
we will
right.
do that. Yes. Okay. Yeah, that's too bad. Yeah. Okay.
Yeah, I think March, pwede siguro you can resend the clusters after all the sessions now. Not only that their field, but also their field of interest. Kasi mukhang maraming nag-excite sa different sessions that we put forth. So again, they may want to re-enlist again. So that when we add more, I think this whole, I mean, we never thought it's gonna be this
exciting.
active. Yeah, I mean, who knew that we can fill up a whole month? It's only Giselle who knew that.
I know. Only Giselle knew that at the beginning. All proven as
It's a little muscling, right? Muscling. Pwersahan ng konti. That's persuasion.
Heavy lifting.
We will have a Christmas edition.
There you go.
Yeah, you'll have a
We will
Zoom
have
Christmas
edition in December.
Yeah, Christmas party for that. Yeah.
I'd like
Good
to,
idea.
I saw Heidi. I haven't had a chance to congratulate Heidi for your paper. Okay?
Yeah, thank you. Thank you, Ma'am Giselle. Thank you also for the support to the Coconut Genomics program.
Milestone. I think it's a major publication. So congratulations.
Thank you.
For leading the effort.
Is this the same site for the rapid
Congratulations to
fire?
the poster.
Yes.
Wendell, I will not be
Thank you,
able to join
participants
the
for rapid fire here.
Minutes.
presentation.
Our
We have
host
a
and
meeting.
chair, Wendell, is here. He's worked so hard for
Yeah,
this.
Wendell is there. I will miss that rapid fire.
Do it.
Congrats, everyone.
It's
Way
amazing.
to go, Wendell.
Yeah.
Wendell. Go ahead, Wendell. Congrats, everyone.
Thank you.
See you.
Thank you.
Thank you, Ma'am
Bye,
Giselle.
Mario.
Bye-bye.
Bye.
Bye.
Yes, so it's really good to give a chance to our members, Marge, to update their clusters or whatever you call
The
it.
other thing I can do is just provide a link, but then I have to, we can have a link to that Excel spreadsheet, like sign in, like we did before, and they can actually just update it themselves. But then I have to invite pretty much everybody to it. Yeah, but alternatively, we can just, if they send me an email, I usually can update right away, and then I'll just post, send to Richmond to post. But the one that I sent to Richmond today is the most updated one that we have. So
maybe if you do just an internal Google Doc first within our group, the Google group, and then everyone has access to it, then yeah, you don't have to go to the website and then put in the password.
yeah, we can put a link to that one like we did with the PowerPoint slide, and then people can just go in. I'm gonna have to give it to Mario because Mario's more better at me at Google Docs. I'm better at OneDrive and the different format. But it would be easy for them to gain access to that one, I can put everybody's email, and then you can just access the folder and then actually update your own data right there in real time. So we can do
Correct.
that.
Yeah, that's a good. That's better.
Yes.
That's work.
Yeah.
That's work for everyone. Yeah.
Without having to email to me, you can just update it there, and then that would be good. Yeah.
Yeah. So when the cluster
Yeah,
decides
I will
to,
do that. I'll give it a try and just list everybody's emails. Yeah.
that would be interesting to see once we break down into sectors and clusters. Who would be the groups?
There would be, somebody can make an analysis of that for each cluster, because you can sign up to 3 clusters, Al.
Yeah.
I wish even 4. I wanted to sign
There,
up
there,
to
there,
4.
there have been people actually telling me that they wish they were in my session, but they're getting another session.
Yeah.
Or I mean, it's like, but it's good competition. There's good variety and choices. So to me, it was not one size fits all. So in a way, they also saw the breadth of our network in that sense. Yeah. So I think it was good. It was all very interesting. Yeah.
Introducing the new members today, wow, it's so impressive. All the credentials, right?
I know,
Yeah, that's a lot of work, guys.
First,
How many members total? Full and
That was 120 for this batch. Yeah.
Wow, that's
a great job, Marge, Mario.
You should go to Mario, because that was the year when we had a lot of these.
Yeah, we made it,
Yeah.
Yeah, we did it.
I mean, as we discussed this morning, if we actually remove the research projects, the research project groups, the RPGs, we can actually expand it so people can be a part of up to 5 different clusters, because there are some people whose interests really go across many areas of research, so we can expand that. Yeah, I mean, you can go from 1 to 5, you have the option, because some people were adding them in the same slot in the Excel spreadsheet, because they wanted to be part of other clusters. So if we remove the RPGs, we can expand the RECs to 5 at least. Yeah.
Perhaps we can remove the RPGs at this point. That was just in our survey. And
kind of redundant, because as long as you have your research expertise defined, that can be used as the classifier for where you want to go.
Right.
We worked with the RPGs because that was what we had from the previous Google survey, but actually the research expertise is a better classifier. Yeah.
Better
and if
classifier.
members themselves decide which cluster they want to be in, that's even better.
And the RPGs now, they're just they would really have to be funded. So there should be a project submission and funding process. So that would originate from clusters, within clusters or intra-cluster. So
and even just Giselle, even just being able to point out where the current funding availability is, and the RFPs coming from different funding agencies is more than enough support for each of these clusters. So I think by them feeling that may pakinabang for them to list in those clusters suddenly, then they really feel the benefit of being a PAASE member. I think that's probably one of the things that we can kind of highlight, us being senior and seeing all the landscape here and then abroad and in the US, of course. You guys, Marge and Edsel and everybody. Yeah, you can also help the young scientists in the US, definitely. But here we can help the Philippine guys here. So I think with the comfort of everyone just getting assembled in Zoom, we don't need to host parties or lunches or spend money traveling. Yeah, now suddenly you can just group them all together and they're all talking already.
So that's what I like to promote tomorrow at the business meeting, that we spend our money for projects and not for get-togethers and food and logistics or things like this. So yes, I think we'll be more focused on the substance of PAASE, the substance of our advocacies. Yeah.
Yeah, even plugging procurement, Giselle, at this point. That's one thing unique about PAASE being not under a government agency like NAS. Because right now I'm looking at the differences between the two. And NAS being under the OST, there's some accountability aspect to it. PAASE, we're a foundation, we're a nonprofit, so we can do a lot of other stuff. So clearly, to me, we can even do a joint private funding, co-funding with private sector to have directed projects like a university industry development programs they do in the US.
So
I mean,
we need work on that. But I think what we need to check is whether if we will be the recipient of huge funds, say here in the Philippines, we must be a foundation accredited.
Correct.
I think we need to get this accreditation, say, from the DOST. So something that we need to look into.
Okay. Yeah, because clearly that's what UPLB Foundation does in procurement for some of the projects in UPLB. They've done very well being able to help in that fashion. I don't know how much. I don't know if Sean is there, but clearly to me, those are looking at the advantages of one private university and foundations in public university like EARThy also. EARThy is funding projects at UP Diliman now because they have members in the alumni that wanted to provide money for COVID.
Yes.
Yeah, they're watching projects with UP Diliman that doesn't have to wait for the OST budget or funding from government. So
that's funding or bridge or funding for preliminary.
Yeah, proof of concept to in order to get a patent, in order to get advanced funding, leading to an SBIR or a DOST grant, or so. That's what Stride did for the longest time. They identified investigators early on who were young, and clearly to me, I think it's now contingent to PAASE to be able to help more of those. The Brian Pajarito, the Hias Junio, the Joyce Ibana 5 years, 7 years ago. The people that you brought in, Giselle, for Balik PhD in UP.
Yeah, these are all Balik PhDs during
Yeah, so I mean, being able to give them grants, now they're more 40, 30, like even Menchi Ablan was now 40, 50 million in grants, and she had the first grants early on 5 years ago.
Well,
so I think that
Balik PhDs in UP got their 3 million pesos, some 4 million pesos startup grants.
But now they're all like 10, 20 million na. I mean, so clearly to me, those are the seeds that we plant. That was that what's Jazza's. I don't know if I forgot which session is it. Is it the FINMA session or is it the NAS session? It says, "What's legacy? Legacy is planting seeds that you'll never see."
And you don't really need recognition. So this is one thing that
doesn't matter.
Really, values much. You don't have to be recognized for it, but you know that you contributed. Okay, so PAASE will not always get recognized for its efforts, but it's okay as long as you know that we're making a major or a
just seeing them grow, Giselle. Exactly. Seeing them grow and bear fruit. That's happiness enough for us.
That's true. Yeah. Okay. Anyway, now I think we have to give Wendell a chance to prepare for the rapid fire competition.
Okay, I'm going to go powder my nose.
Powder your nose. These are our future leaders. Our finalists.
9:30.
Yeah, 9:30. So am I right?
Okay.
Maybe it's 9:24. So maybe Wendell, you have some last minute advice to our finalists. So take it away, Wendell.
Okay, thank you very much, Giselle.
Yes. Thanks. I'm going to stay, of course.
Okay. Good morning once again, everyone. Welcome to the rapid fire competition during the 40th APAMS of the PAASE. As you have heard earlier, we have actually experienced a lot of first times in this scientific meeting. And for the very first time also, we are doing this rapid fire competition of selected poster presenters via Zoom. So we hope for the success of this competition. Now, just to give you a brief background about this rapid fire competition, Of course, you know that we had a call for abstracts, and around 45 abstracts were actually submitted. All the 45 abstracts were of good quality, so we all invited them to submit their posters. But of the 45, 41 only submitted their posters. And of the 41, 35 indicated their interest to compete for the rapid fire. Well, of course, that was a big number for a competition. So what we did was to, of course, invite members of the Board of Judges to finally select the finalists for this competition. Okay, 21 were actually selected by the Board of Judges to present for this for today's competition. I hope that you were able to visit the PAASE website the past days. We had the poster presentations on August 6, August 7, 10, and also 11. The 41 posters were actually distributed into 4 clusters, and they were uploaded into our PAASE website. On August 6, we had the Ecology and Marine Science poster presentation. On August 7, we had the Plant Science and Agricultural cluster. For August 10, we had the Material Science and Technology cluster. And yesterday, we had the Disease, Medicine, and Pharmaceutical Science cluster. Now, let me inform you of the criteria used by the judges in selecting all our finalists today. Okay, the criteria are 85% for the quality of scientific objectives, study design, methods, data, results, and conclusion. And then 15% for the quality of presentation, visual and textual. Now, how about the criteria that will be used by the judges today? Okay, there's a slight change in the criteria. 70% for the quality of scientific objectives, study design, methods, data, results, and conclusion. 20% for the quality of oral delivery of our finalists, and 10% for the quality of visual materials. So let us congratulate all our finalists today, 21 of them. Now, the composition is very interesting. Majority are actually graduate students. Some of them are research associates and junior faculty members of universities. Now, the Material Science and Technology cluster is unique. Mostly are established researchers from the Philippine Textile Research Institute of the Department of Science and Technology and a faculty member of University of Santo Tomas. So the level is a little bit different from the other clusters. Of course, it's appropriate for us to recognize the members of the Board of Judges who worked hard to read and also, of course, evaluate the posters. And they will once again evaluate the finalists and choose the top 3 for today's competition for each cluster. Okay, so as I call the name, maybe the member of the Board of Judges can actually open your video and also your turn on your audio so you can give a brief greeting. Okay, for the Ecology and Marine Science cluster, we have with us Dr. Veronica Migo, Professor of the Department of Chemical Engineering, College of Engineering and Agrotechnology of UP Los Baños.
Hello, everyone. Good morning.
Morning, ma'am. Thank you. Then we have Dr. Teresita Perez, Professorial Lecturer of the Institute of Biology, UP Diliman.
Hello, everyone. Good morning.
Thank you, ma'am. Dr. Irene Rodriguez, who serves as the chair of the Board of Judges for Ecology and Marine Science. She's an associate professor of the Marine Science Institute, UP Diliman.
Good morning, everyone.
Thank you, Irene. Now let's move to the Board of Judges for Plant Science and Agriculture. We have Dr. Victor Amoroso, University Professor of the Central Mindanao University. Okay, so Dr. Vic is around, I think. Okay, and then we have Dr. Heidi Galvez, Associate Professor of the Institute of Crop Science, UP Los Baños.
Good morning, everyone.
Thank you, Heidi. and the Chair of the Board of Judges for Plant Science and Agriculture, Dr. Lilian Rodriguez, Assistant Professor of the Institute of Biology, UP Diliman.
Morning po.
Thank you, Lilian. Now let's move to the Material Science and Technology cluster. First, we have Dr. Marlon Conato, Professor of the Institute of Chemistry, UP Diliman.
Hi po, good morning.
Thank you, Marlon. Dr. Elmer Estacio, who serves as the chair for this cluster. He's a professor of physics in the National Institute of Physics, UP Diliman.
Morning po, ng umaga. Hi, sir.
Thank you, Tux. And lastly, we have Dr. Joey Ocon, Associate Professor of the Department of Chemical Engineering, UP Diliman.
Morning po.
Thank you, Joey. Now let's move to the last cluster, Disease, Medicine, and Pharmaceutical Science. First, we have Dr. Leslie Michelle Dalmasio, Professor of the Department of Biochemistry and Molecular Biology, College of Medicine, UP Manila.
Good morning, everyone.
Thank you, Les. The Chair of the Board of Judges for Disease, Medicine, and Pharmaceutical Science, Dr. Philip Ian Padilla, Professor of the Division of Biological Sciences, UP Visayas.
Hi, everyone. Good morning.
Hi, Philip. And last but not the least, we have Dr. Michael Velarde, Professor of the Institute of Biology, UP Diliman.
Hello, everyone.
Hi, Mike. Okay, thank you very much, members of the Board of Judges. They worked very hard for this competition. And as you have noted, we have a good pool of the Board of Judges. So we expect, of course, a good decision to be made for this competition. Now, a few guidelines for today's presentation of the finalists. Each finalist will deliver his or her presentation in 3 minutes only, using 3 PowerPoint slides. There will be no question and answer part, no Q&A. Although the judges have evaluated the posters, gave them scores, and eventually ranked them, this session is a fresh start. Finalists' scores are actually back to 0, and they need to earn their new scores based on the criteria I mentioned earlier. Again, each presentation should last for 3 minutes only. And this will be strictly implemented. How are we going to do that? Okay, a presenter will be muted by the host if you exceed this allotted time. Of course, there would be glitches. We will make room for that. Okay, but the slides that you have actually submitted to me will be managed by the host. So please inform the host if he needs to move your animation or your slide. Okay, so without further ado, we would like to begin our competition. We have 21 finalists again, and let's start with the first one. Our first finalist is in the Ecology and Marine Science cluster from the University of San Agustin, Iloilo. Let's have the presentation of Ms. Edna Sabido.
Good morning. Good morning, everyone. My name is Edna Sabido of CMG3, University of San Agustin, Iloilo, and I will present our work entitled Exploring the Diversity and Antibiotic Potential of Iloilo Marine Sediment-Derived Actinobacteria using multi-locus sequence analysis, metabolomics, and bioassay-based strategies. Next, please. This study aims to investigate the diversity of actinobacteria from marine sediments of Iloilo and to find antibiotic leads against multi-drug-resistant pathogens. Next, please. To achieve this goal, we employed multidisciplinary approach using techniques in microbiology, genomics, metabolomics, and natural product chemistry. Next, please. We isolated 17 bacterial isolates that belong to 3 genera: Glycomyces, Nocardiopsis, and Streptomyces as a major cluster. However, the diversity index is only 0.044, suggesting low genetic diversity. Next, please. The Streptomyces isolates in the phylogenetic tree do not have a distinct clade. Therefore, we elucidated the relatedness of the Streptomyces species by multi-locus sequence analysis that yielded a more resolved separation of the species. Next, please. Notably, streptomyces strain DSD040 has separated itself from the group, suggesting a slight genetic variation. Next, please. The antibacterial screening showed that 14 isolates inhibited the growth of the multi-drug resistant strain of Staphylococcus aureus or MRSA. Next, please. And based on our preliminary results, we focused on specific streptomyces strain, which is DSD011, showing the highest growth inhibition against MRSA. Next, please. Then its crude extract was purified using different chromatographic techniques, which was guided by antibacterial assay until a pure fraction was obtained with 98% growth inhibition against MRSA. Next, please. The pure bioactive compound was analyzed by mass spectrometry, revealing a mass of 597. Next, please. The MSMS and the GNPS analysis showed that the compound, next please, was corresponded to fidomycin D, which is an anthracycline glycoside, and this is the first antibacterial activity report for fidomycin D against MRSA. Next, please. And in conclusion, our library of marine sediment-derived actinobacteria from Iloilo belongs to the phylum Actinobacteria of diverse ancestral origin, and they produce secondary metabolites that may serve as a potential antibiotic leads. Next, please. To end, I would like to thank our LGU partners and funding agency, which is the PCHRD DOST, and for your attention. Thank you very much.
Thank you very much, Ms. Sabido. Okay, now let's move to the second finalist of the same cluster, Ms. Dana Joanne Von Trono, also from the University of San Agustin, Iloilo.
Good morning, everyone. My name is Dana Trono from Sito Vito University of San Agustin. Our work is entitled Multiple Strain Analysis Reveals Intra-Species Heterogeneity in Antibiotic Activity of Streptomyces species from Philippine Marine Sediment. Now, Streptomyces strains of the same species are not genetically identical and produce various secondary metabolites. Yet, it remains unclear whether this results in differences in their bioactivity. Thus, we aim to profile the diversity and bioactivity of phylogenetically identical Streptomyces strains by addressing the specific objectives shown on the slide. To achieve this, we collected marine sediments from different sites in the Philippines and isolated actinobacteria using culture-dependent techniques, followed by antibiotic screening. Now, extracts with more than 50% bioactivity were analyzed for molecular identification. Next, please. From 15 sampling sites, 2,780 Streptomyces strains were isolated and 134 strains have confirmed with antibiotic activity. Next, please. These active isolates belongs to 20 Streptomyces species. Notably, we isolated 3 novel antibiotic-producing Streptomyces species as indicated on the slide. Next, please. Our study revealed that the two Bataha reefs, southeastern Iloilo, southern Antique, as the most diverse sites. Moreover, Streptomyces parvulus, rochei, and Anisocaecilis are the most abundant and ubiquitous in all sampling sites. Next, please. The heat map of antibacterial activity of parvulus strains showed that some have strong activity as indicated in red, others have moderate activity, while some are inactive as shown in black. Interestingly, identical strains isolated from the same site still have varying antibiotic strength. Next, please. These identical strains have similar set of PKS1 and 2 domains. This means that the PKS genes are highly conserved in closely related strains and do not dictate the antibiotic variation we observe in our study. Instead, the presence of strain-specific genes and cryptic ecological niches may have influenced the antibiotic variation as reported by other studies. Next, please. In conclusion, this is the first large-scale study to demonstrate the strain-level antibiotic variation within Streptomyces species, which emphasizes the importance of sampling identical strains for bioprospection. And thank you very much for your kind attention.
Thank you very much, Ms. Trono. Now let's move to the third finalist of this cluster. We have Ms. May Ashley Nacario of the Institute of Biology, UP Diliman.
So good morning, everyone. My name is May Ashley Nacario, and I will be presenting our poster entitled DNA Fingerprinting Using Box A1R and GTG5 Primers to identify sources of fecal contamination in selected sites and tributaries of Laguna Lake. So may I request the host to click the presentation up to the last object of the first slide, please? Thank you. So just to give you a brief background of our study, our research is focused on Laguna Lake, which is considered as an important freshwater lake in the Philippines. However, the water quality of the lake is slowly deteriorating. Wastewater discharged into the lake may contain harmful microorganisms like E. coli and other disease-causing microbes. This causes health risks to the estimated 16 million residents along the lake's vicinity. Next, please. So the main objective of this study is to identify the dominant source of fecal contamination in Laguna Lake and 8 of its most polluted tributaries. Next slide, please. So to achieve this goal, next, we followed this scheme for our methodology. And what we basically did was to construct a host origin library composed of box A1R and GTG5 fingerprints from E. coli isolated from different host sources. And then we tested the performance and accuracy of our library using discriminant analysis of principal components or DAPC. And lastly, we did microbial source tracking of contamination using DNA fingerprints isolated from the lake and the 8 tributaries. So what we found out is that the host origin library that we constructed has a relatively high accuracy of 84.82%, which means that it can be used for microbial source tracking or MST. Next slide. And so using this library to the MST in Laguna Lake and the 8 tributaries, we found out that most sites were dominated by sewage contamination for both the wet and the dry seasons, as shown in this figure. Next. We therefore conclude that, next please. Various sources dominated by domestic sewage contaminate the lake and some of its tributaries. And also, the major source of contamination may vary depending on the season. And for the future directions of this study, these findings can serve as science-based information for policymaking to prevent the degradation of Laguna Lake. And ultimately, next please. It is also recommended to apply MST studies on other water bodies, especially to the ones that need rehabilitation and water quality improvement. That's all. Thank you.
Thank you very much, Ms. Nacario. Now let's move to the next one, Ms. Nicole Bolo of the Institute of Biology, UP Diliman.
Good morning, everyone. I'm Nicole Bola, and I will be presenting our study. Next, please. Next. Antibiotics are used to treat bacterial infections. Thus, they serve as a cornerstone of modern medicine. Next. However, due to misuse, high amounts end up in the environment. These become hotspots for resistance evolution. Next. These resistance genes can be shared between bacterial cells. Next. Then, these resistant strains can spread among humans and animals via consumption or contact. Next. Eventually, this will render antibiotics ineffective. Next. With this problem, our aim was to investigate the occurrence of resistant E. coli and determine their multiple antibiotic resistance indices. The results of the study can increase public awareness about antibiotic resistance and can lead to the improvement of water quality of Laguna Lake. Next. To accomplish these aims, we collected water and fecal-associated samples. Then, we adapted the US EPA membrane filtration protocol to isolate E. coli. Lastly, we used Vitec-2 for the antibiotic resistance assay. Next, please. Next. From the 450 isolates, varying resistance against 14 antibiotics was observed. Alarmingly, 180 of these isolates showed resistance against ampicillin. Next. A significant difference in the resistance of isolates was observed among fecal samples. Most notably, chicken isolates showed the highest occurrence of resistance. Next. Furthermore, chicken isolates also rendered the highest MAR index, meaning some isolates are resistant to more than one antibiotics. This may be due to the fact that in the poultry industry, antibiotics are sometimes used to prevent diseases. This defeats the purpose of antibiotics, which are meant to treat existing bacterial illnesses, not prevent them. Next. So, in conclusion, we were able to isolate antibiotic-resistant E. coli. Among fecal samples, chicken isolates showed the highest occurrence of resistance and the highest MAR index. Next. So, what can we do to prevent the spread of antibiotic resistance? One way is to avoid indiscriminate use of antibiotics for viral illnesses, such as for flu or colds. Also, if we take antibiotics, we must adhere strictly to the doctor's prescription in terms of dosage and duration. Next. With the current situation, it is not a matter of if, but a matter of when the superbugs take over. We can only hope that by then we would be equipped against them. Next, please. For more information, you may visit this site. Thank you for your attention.
Thank you very much, Ms. Bolo. Now let's move to the last finalist of this cluster. We have Ms. Jamaica Caras from the Institute of Biology, UP Diliman.
Ms. Caras, you may begin. Hello,
Hello.
Ms. Caras.
Oh, sorry, my internet was unstable. Good day, everyone. On behalf of my co-authors, I will be presenting to you the details of our study, bacteriological and histological findings in cetaceans that stranded in the Philippines from 2017 to 2018. The Philippines is a host to a diverse assemblage of 29 species, representing one-third of the world's cetacean diversity. Every year, around 100 cetaceans are stranded in beaches all over the country. Various factors such as environmental conditions, acoustic trauma, or probably a disease can be attributed to these strandings. Next slide, please. In collaboration with the Philippine Marine Mammal Stranding Network, a rescue response and/or
Hello, Ms. Caras, are you still with us?
in collaboration with the Philippine Marine Mammal Stranding Network, a rescue response and/or a rehabilitation is conducted in an attempt to save the animal. In an unfortunate event that the animal dies, a necropsy is conducted and samples such as blood, tissues, and swabs are collected and are sent to UP Diliman for storage and processing of samples. In the years 2017 to 2018, biological samples were collected from 21 individual cetaceans. The samples were subjected to histological assessment and antibiotic resistance screening. Next slide, please. Sorry, I'll try to finish my time. Fixed sample tissues were stained with hematoxylin and eosine and were observed under light microscopy. Lesions that were observed in tissues include congestion, hemorrhage, edema, hemosiderosis, glomerulopathy, Zenker's necrosis, atrophy, atelectasis, and parasitic cysts. These lesions may be associated with factors possibly contributing to death, debility, and stress of animals during their stranding. On the other hand, with the use of Kirby-Bauer assay, the resistance of 24 bacteria were tested against 18 antibiotics. All 24 isolates were resistant to at least one antibiotic class, and 79.17% were classified as multiple antibiotic resistant, or MAR. The MAR index of the isolates
I think Ms. Caras is having network difficulties, right?
Can I still continue?
Yes, you still have 20 seconds.
Okay. The isolates were most resistant to erythromycin and most susceptible to immunem. Knowing the antibiotic resistance of common bacteria in cetaceans can help veterinarians in decision-making with regards to the animal's rehabilitation. Information on pathologies in populations on cetaceans caused by biological agents are urgently needed, as these may provide clues on how to develop effective conservation strategies to preserve the trophic structure of the marine ecosystem. As cetaceans are difficult to observe and sample, the information generated from the stranded individuals are valuable to evaluate the impacts of anthropogenic activities such as fisheries interaction and land sea movement of biological pollutants. That's all, thank you, and apologies for the many interruptions.
We understand.
Thank you very much, Ms. Caras. Okay, now let's move to the plant science and agriculture cluster. Our first presenter for this cluster is Mr. Jose Arnel Reyes of the Institute of Plant Breeding, UP Los Baños.
Okay, thank you. Good morning. I'm Jose Arnel Reyes from UPLB, and I conducted this mini research last year under the supervision of Dr. Soto. So yield improvement has always been one of the main objectives of a plant breeder. On an economic point of view, yield not only dictates the amount of profit a farmer can earn, but also the number of people one can feed. So this is a very important trait. However, yield is not easy to improve. It's a function of crop genetic makeup and its environment, as well as the interaction of the two. One of the steps in crop development is the proper identification of plant traits that could result into higher yield. But sometimes there are characters that are overlooked or not yet fully utilized, even though many studies suggest its direct correlation with yield. And these are the traits coming from below the ground, the root system. Next slide, please. Thus, in order to see the significance of the root system in the dry matter accumulation, we tested the effect of exogenous application of auxin to mung bean root growth. Auxin is a phytohormone naturally occurring in plants and considered as a key regulator of root growth. Proper root growth during the vegetative stages has showed positive effects in terms of water and nutrient absorption. So first, we treat the mung bean seeds with different concentrations of indole triatic acid and auxin by soaking and then pre-germinating it before sowing it the following day. Root sampling and dry matter data were gathered at early vegetative stages, which were analysis was done using analysis software Winerizo. Next slide, please. Results revealed a particular trend across different root parameters. Root length, root surface area, and root volume were all improved in the addition of 0.01 ppm IAA, but at higher concentrations like 10 ppm, significant reduction in growth was observed. These were all expected as higher levels of auxin could induce growth retardation in plants. Additionally, plants with the best root growth accumulated the highest dry matter. So what do all these suggest? Well, for one, it shows the optimal concentrations of IAAA could induce root growth in mung bean and possibly improve its yield potential. The possibility to identify other sources of IAA or any type of auxin and utilize it to improve crop production could be further investigated. Yes, it's a simple experiment, but it provided information on the possible uses of auxin root growth and ultimately in crop yield improvement. So that's all. Thank you.
Thank you very much, Mr. Reyes. Let's move to the next one, Mr. Arnel Celestino of the Graduate School of UP Los Baños.
Yes, good day to all. I am Arnel B. Celestino. I will be presenting a part of my thesis which focuses on the morphoanatomical responses and alleviation of waterlogging stress in onion. My objectives are to determine the effect of the waterlogging stress on the morphoanatomical traits of onion, then investigate the effectiveness of the fish amino acid fertilizer and potassium silicate in alleviating the harmful effects of the waterlogging stress. For the methodology, cultivation of red dragon onion in this research followed the onion production guide by the Department of Agriculture. Then the pot experiment under greenhouse condition was arranged in strip split plot block design. Then the waterlogging imposed artificially, and the recommended rate of the commercial FAA and potassium silicate is foliarly applied. Then the root cross sections and leaf epidermal peelings were sampled for root characterization and stomatal responses. Then the data were analyzed using ANOVA Tukey's test. Next slide, please. For the results and discussion, the number of adventitious root during the bulbing stage was significantly higher than the pre-bulbing, and more adventitious root but fewer secondary lateral branching of roots were observed in all waterlogged onions, whether it is treated with FAA, potassium silicate, and untreated. So we can see the picture below, the adventitious root and the secondary lateral branching. Then presence of suberized exodermis, as we can see at the picture below. The red stain in the exodermis, it serves as the protective barrier, which restricts the water movement, reduces internal oxygen loss, and prevent the solute leakage. Next slide, please. Then stomatal responses, the waterlogging stress affected the stomatal development and processes and induced stomatal closure, which we can see the decline of stomatal density and stomatal aperture in the waterlogged onions untreated. And also the limited facilitation of water uptake and exchange of gases through partial stomatal regulation, we can see the higher stomatal density and aperture in FAA and potassium silicate treated in onions, which alleviated the effects of the water stress. And for the conclusions, the application of FAA and potassium silicate in waterlogged onions increase the stomatal density and stomatal aperture, and waterlogged onions develop new adventitious roots and form suberin layer in exodermis. And also, the quantitative analysis of root architecture and suberin accumulation is recommended. And also, this research is for additional for cultural practice and management for the farmers to reduce crop loss. Thank you.
Thank you very much, Mr. Celestino. Now let's move on to the next finalist, Ralph Allen Geronimo of the Department of Biology, Ateneo de Manila University.
Good morning and good evening to everyone. I'm Ralph Allen Geronimo from Ateneo de Manila University. Our research is about microbiological evaluation of virgin coconut oil production in the Philippines. This study is part of the research program by Dr. Fabian Dairis, aims to revise the Philippine National Standard for VCO. The Philippines, next please. The Philippines is one of the leading exporters of VCO in the world. However, there is lack of studies regarding the microbiology of the VCO production. Next. Therefore, this research aims to determine the microbial quality and safety of VCO during production. Next po. In order to do that, we have visited 9 VCO producers from all across the country. We collected samples from each production step and from their production line and tested them for several microbial parameters. Next. This figure shows the process flow and samples collected from producer E2, just one of the producers listed. The process flow varied greatly from different VCO producers. Next, please. This table shows the microbial load samples from that particular producer. We collected samples from 2 production lines. Next po. It was observed that aerobic plate count, yeast and mold count, and coliform counts were highest at the beginning of the production line. Next po. Next po. E. coli was detected in 2 samples from producer E2. SRUs, E. coli, and Salmonella were also detected in several samples from the other VCO producers. Next po. Next po. Yeah. Both the finished products from producer E2 failed the Philippine National Standard for VCO. In fact, only 3 out of the 18 finished products tested passed the Philippine National Standard for VCO. They typically failed the APC and YMC standards. Contamination may have come from the workers or from the biofilm formation in the equipment. This means that the producers should follow good manufacturing practices to ensure production of high quality and safe VCO with microbial counts that pass the PNS for VCO. This research, the results from this study may be used as a basis for the revision of the PNS for VCO. That's all. Thank you for listening.
Thank you very much, Mr. Geronimo. Now let's move on to the next one, Mr. Jeff Ofornia of Cagayan State University.
Good morning, teaching everyone. Today, I will be presenting a research entitled Layer of Sclerenchyma band as an Anatomical Hindrance to the Adventitious Rooting of Hard-to-Root Excelsa coffee Stem Cuttings. This study was conducted to uncover the reason why Excelsa coffee is hard to root when propagated asexually. So basically, this study was conducted for using histological procedure, using paraffin microtome and slide preparation. The microscope slides were viewed using acioscope microscope, and the stem anatomy and structures were investigated. We also rooted the coffee stem cuttings by which basal incisions were made on the rooting zone to break the sclerenchyma band present in the stem cuttings. Next slide, please. Based on our result in figure 1, we have measured the thickness of the stem structures of both species of coffee, and we have found out that Excelsa coffee has thicker stem structures as compared to Robusta coffee. However, the most probable anatomical difference why Excelsa coffee is harder to root as compared to Robusta coffee is the presence of narrow, compact, clustered ends, nearly continuous layer of sclerenchyma band as opposed to the discontinuous sclerenchyma band in Robusta coffee. The layer of sclerenchyma band in Excelsa coffee may prevented the protrusion of roots in the stem cuttings. Moreover, as the Excelsa coffee and Robusta coffee were propagated for 4 months, the continuous layer of sclerenchyma band in Excelsa coffee produced gaps which may serve as signal for possible root initiation in the stem cuttings, which is found to be similar in Robusta coffee. Next slide, please. Next slide, please. And to facilitate breaking of the layer of sclerenchyma band in Excelsa coffee, basal incisions were made in the rooting zone of the Excelsa coffee stem cuttings. And this promoted more adventitious roots to initiate in the Excelsa coffee stem cuttings. For future studies, a combination of root-promoting chemicals may be applied after making several incision cuts on the rooting zone of the stem cuttings to facilitate earlier rooting and to increase the root number of the hard-to-root Excelsa coffee stem cuttings. That's all. Thank you.
Thank you very much, Mr. Opeña. Now let's move to the last finalist for this cluster. We have Mr. Russell Van Roa from the Graduate School of UP Los Baños.
So a pleasant morning to everyone. So let me present to you our study, which is entitled In Vitro Responses of Lettuce Genotypes to Varying Levels of Benzylaminopirin. So next, please. Next. The success of crop improvement through biotechnology requires a reliable and efficient in vitro regeneration system. As per our crop of interest, which is lettuce, the ability to regenerate in vitro usually depends on the genotype and the culture environment. So in addition for that, a regeneration system that induces production of multiple shoots is desired. So to address this issue, we conducted this study to examine responses of 4 different lettuce genotypes, namely Carlo Rosa, Emperor, Grand Rapids, and Green Towers. They were cultured in a suitable media treated with a known plant growth promoting hormone cytokinin, which is in a form of benzylaminopirin that comes in different concentrations. Next, please. Next. Next. So we did this experiment by culturing cotyledons from in vitro grown 3-day-old seedlings in a suitable medium given with 0, 5, and 10 milligram per liter of benzylaminopirin. So after 20 days of culture, interestingly, we found that fresh weights were significantly improved in Grand Rapids as well as in Green Towers. So this explains that there are some growth that happen on the cultured cotyledons. So next, please. Another more interesting finding is that at high levels of BA, cotyledon X plant produced multiple shoots except for Emperor. On the other hand, next please. With low to no BA, root production was stimulated. So next please. Next. Okay. To show you better the responses, I present to you these figures as a sort of evidence. So we have X plants producing roots as shown in letter A. In letter B, we have roots coming from callus. So we have letter C, which has produced callus. And most interesting part is the production of multiple shoots as seen in letter F. So next, please. Now, next. So now, with the results obtained, it is now enough to say that high BA promotes the production of shoots, although genotype dependent. And in contrast, at low to no BA, it stimulates the production of roots. And the grand winner for this research is Grand Rapids, in which it performs best in vitro by producing a lot of multiple shoots. So that will be all. Thank you.
Thank you very much, Mr. Roa. Now let's move to the presentation of the finalists of the Material Science and Technology cluster. Our first presenter is Rachel Sapin of the University of Santo Tomas.
Good morning. It is my honor to present to you our study entitled Abaca Cotton Fabric with Multi-Walled Carbon Nanotubes and Polyaniline Polypyrrole Complex Layer, a greener approach to energy storage. Next, please. Supercapacitors are energy storage devices that bridge the gap between conventional capacitors and batteries, and its most essential part is the electrode, as it dictates the mechanism of electron transport as well as the amount of energy that it could store and release. Usually, support material for supercapacitor electrodes are based on consumable materials such as carbon paper, metal sheets, and the like. But for this study, we wanted to use indigenous fabric blended with cotton to highlight the sustainability of our electrode. Next, please. We have chosen to use Abaca cotton fabric, multi-walled carbon nanotubes, and polyaniline polypyrrole complex layer since their individual characteristics fall under the ideal supercapacitor electrode properties, and together they make up for each other's disadvantages and further improve the electrode's capacitive activity. Next, please. In this study, we aim to prepare a PAPI MWCNT ACF fabric supercapacitor and to characterize it according to its physical and electrochemical properties, as well as to apply the fabricated supercapacitor to an LED sample. Next, please. So this is the photo of the actual fabricated supercapacitor electrode. Next. And these are the supercapacitor cells connected in series to light the LED. Double click, please. Looking at the table, PAPI MWCNT ACF generated the lowest equivalent series resistance and the highest electrochemical values among the three composites due to the additional network in the complex polymer layer. Next, please. For the LED testing, a positive net charge discharge was obtained for the fabricated supercapacitor, making it capable of energy storage and delivery. Next, please. So to sum it all up, our study showed that PAPI, MWC, and ACF exhibited improved electrochemical values, thus better capacitive behavior, and its power and energy density values fall well within the supercapacitor range in the Ragone plot, indicating that it can be an ideal supercapacitor electrode material. Now, to power up a variety of low-powered devices, a solar panel may be used as a power source instead of a battery, to further emphasize its application as a renewable and sustainable energy storage device. Thank you.
Thank you very much, Ms. Kausapi. Now let's move to the next finalist, Mr. Agustin Bordallo Jr. of the PTRI DOST.
Good morning, everyone. I am Agustin Bordallo Jr. of DOST PTRI, presenting our research on varietal locational variability of Philippine pineapple Ananas squamosus leaf fiber fineness for Spaniard processing. Pineapple is the most expensive natural plant-based textile fiber in the Philippines today. Traditionally, pineapple fiber for textile has been used extensively in Aklan using the red Spanish variety of the Ananas family. However, its application for a more industrial textile manufacturing has made it a sought-after fiber for the production of not only fabrics but also leather-like material for designer bags and shoes, and this time using plantation pineapple varieties like Hawaiian and Formosa. The Philippines has a total of 66,000 hectares of pineapple plantation, which make it a sustainable source of raw material. And amongst the top 5 with the largest area of pineapple plantation is Bicol, Davao, and Calabarzon region. Initially, only Camarines Norte or the Bicol region actively produces fiber for textile application, but the demand and opportunities has already rippled to some major producers of pineapple in the country. The study therefore aims to determine if varietal and geographical location affects the performance of fiber processing, specifically for yarn production. The study evaluated the fiber's properties in terms of chemophysical properties and processing performance. Next slide, please. Next slide, please. The fiber treatment or degamming was conducted following the patented treatment procedure established by PTRI to render the material pliable for yarn processing. The treated pineapple material was then cut, opened, and blended with a carrier of 75% cotton before subjecting the lab to the laboratory's yarn of spinning. Fiber properties of raw and treated fibers and yarn spinning performance were evaluated and analyzed for any varietal or geographical dependence. Next slide, please. Next slide, please. Percent fiber recovery and percent total cellulose was determined using raw and treated pineapple. Cellulose is regarded as a textile polymer, and generally, the higher the cellulose content, the more resilient the fiber is in chemical and mechanical processing. From the experiment conducted, F200 pineapple variety was the highest total cellulose content, while Formosa from Camarines Sur has the lowest total cellulose content. During yarn spinning, the performance of F200 variety from Davao was also recorded to have the lowest ends down or stoppage due to breakage, which is one of the critical parameters in evaluating this performance. Next slide. To conclude, F200 pineapple Davao variety was observed to have the highest total cellulose content and the best spinning performance amongst the three fibers studied. However, the project team recognized that to determine whether the effect was due to geographical or varietal difference, same varieties must be gathered from all locations. In addition, since agricultural crops are heavily dependent in different seasons, the timing when the samples are collected must also be kept the same to limit the effect of season changes in the fiber properties. This direction can be pursued in future studies. Thank you very much for the attention given.
Thank you very much, Mr. Bordallo. Let's move on to the next one, Mr. Cornelio Loyola of the PTRI DOST.
Hi, good morning, everyone. Can I please ask the host to click on the animation until the last animation for this slide? Pinoy pride has seen resurgence over the past few years. Our very own Miss Universe Catriona Gray showcases the beauty and story of our own Filipino traditional textiles. The narrative, the know-how, and the patterns are bound to oblivion as our women, the repository of our textile heritage, are advancing in age, and the blueprints of these textiles, including its space, have not been brought up to speed. In PTRI, we do not only develop textile materials, processes, products, services, but we also engage in preservation of textile knowledge and use these to appropriate and to inspire product development. For the objective of the study, it is to digitize traditional Filipino weaves using WeavePoint, a software employed in weaving to analyze, store, and generate patterns. For the methodology, it evaluated and analyzed the binocol patterns of Ilocos, Hablan pattern of Iloilo, and Inol pattern of Maguindanao, and generate a manual draft that can be stored and communicated. The selection of the weave patterns for this study centered on the three groups of the archipelago. These were executed with the WeavePoint software, and the digital rendition of the patterns was created, and this will be the blueprint of the study. Next slide, please. The traditional process, as illustrated in Figure 1, next, entails 8 major activities in creating a single weave of fabric. It can take as much as 2 days for preparation, and the succeeding days were devoted to the weaving proper. A day is equivalent to 8 hours to produce 7 meters of fabric. The digital rendition, as seen in Figure 2, of the selected fabric with the aid of WeavePoint has proven its ability to simulate the design fabric prior to execution. Next. The topmost part will be the warp or the lateral threads next, and the leftmost part, or the rightmost part is where the weft or the filling of the fabric is. Next, 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 and next. This led into a digital rendition using the software, which resulted to an elevated, cost-efficient product development wherein the total time has been cut significantly, up to 50% from the traditional process, with the minimal chance of unfavorable outcome. Next. Moreover, the execution of the manual draft pattern in the bundled computerized Dolby Loom machine, as in this figure, can now be executed with a fabric up to 60-inch width, which is seen as an advantage for design innovation. Last slide, please. For the future plans, being able to initiate a catalog, define its origin and narrative, and create a digital database of the traditional Filipino textiles would elevate our textiles, as the availability of the pattern design details will be stored, protected, appropriated, and accessible. Next, on a product development level, creation of a digital rendition with the use of a CAD software will not only digitally store these treasures, but also orient our traditional textiles to Industry 4.0. Next, this will actually give a uniquely Filipino stamp for the DNA. Thank you for listening.
Thank you very much, Mr. Loyola. Our next presenter is Roberto Casero, also from the PTRI DOST.
Yes, good morning. I am Roberto Casero of the Philippine Textile Research Institute. Our work is about thermoacoustic nonwoven textiles from Abaca Musa Textilis knee fibers. For the background, next please. Next please. Yarns or fibers can be converted into fabric depending on the materials and methods employed. Most fabrics are produced by the method of interlacing, such as, next please, knitting, which is made up of a single yarn looped continuously to produce a braided look, weaving, which is made up of multiple yarns crossing each other at right angles to form the grain, and nonwoven, which is a web structure bonded together by entangling mechanically, thermally, or chemically. Abaca undeniably has more advantages over synthetic fibers. Not only are they environment friendly, which is the world's priority at this time, but more importantly, they are sustainable and biodegradable. And to date, the Philippines has more than 72,000 Abaca plantation and is the top exporter of Abaca fiber to the world, which accounts for 87%. It is the objective of the project to diversify the application of natural fiber-based nonwovens for insulation, and it will offer a unique material and niche product for nonwovens. Next slide, please. Next, please. Next, please. Yes. Abaca pseudo stems were decorticated using the decorticating machine to extract the fibers. The fibers were then degummed with low alkali followed by one bath scar bleaching. Next, please. The dried fibers were softened using the mechanical softening machine. And next, please. Cut into 2 inches staples before opening. The cut fibers were blended with polyester fibers using a 50-50 blend ratio. The blended fibers were processed using the mini carding machine for lap formation and were then converted to nonwoven fabric using the needle punching machine with fabric weight of 150 and 300 grams per square meter. Next slide, please. For its performance, looking at table 1, the breaking strength was higher but lower elongation in the machine direction. Next, please. This is attributed to relatively more oriented fibers at machine direction, thus improving the strength, and more random fibers in cross machine direction, thus improving the elasticity of the material. Fabrics along MD have lower values of air permeability because the arrangement of fibers in parallel laid fabrics makes the fabric structure more compact, so it offers more resistance to the flow of air. The thermal conductivity of the nonwoven fabric is 0.0358 watt per meter Kelvin, which is comparable with commercial glass wool and foam thermal insulation. The sound transmission loss of the Abaca nonwoven fabric was 2 decibels at 500 hertz, which was comparable to that of commercial acoustic insulation. Looking at the right side of the slide are the prototype insulating materials developed from the Abaca polyester nonwoven fabrics. And lastly, Abaca nonwoven textiles as thermoacoustic material is part of the nonwoven program, which will not only be used for wearables, but for building and smart installation as well. Thank you for listening.
Thank you very much, Mr. Casero. And moving on, let's have Ms. Cheryl Lopez of the PTRI DOST.
Good day from DOST PTRI. May I have the privilege to present to you the study entitled Commercialization of Bivoltin Bombix Moriel Silkworm Hybrid Cocoon Production in Support of Philippine Silk Industry. Since sericulture or the rearing of silkworms of silk fibers is not endemic in the Philippines, the country used to heavily depend on importation of hybrid eggs from leading sericulture countries in Asia. Due to the long travel, a very low hatching rate became the biggest constraint, where egg mortality is reach to as high as 90%, leaving a very small percentage of larvae to grow. PTRI's responsive contribution to the silk industry in the Philippines, can you go back to the previous slide, please? Led to the R&D for better silkworm hybrids that would be at least locally available and disease-free. So over a period of time, breeding activities were able to develop hybrids that have become suitable to the local conditions. Particularly, 4 hybrids were developed in Mindanao. Now, after years of rearing trials, PTRI was able to come up with a package of sericulture technologies that have become useful in the fields of cocoon producers. Next slide, please. Silk is such a beautiful natural fiber material. PTRI values its excellence by ensuring that hybrid lines come from quality silkworm parent combiners that will bring about higher productivity. A breakthrough, PTRI succeeded in developing 4 new hybrids that have significantly reached hatching percentage to 99%. Robust and healthy larvae, they have produced silk cocoons that become pivotal in the commercial use of hybrids. In total, just 57% of silk cocoons produced Class A grade, and also the hybrids received the highest 5A reliability grade, indicating their efficiency and good silk recovery in the reeling process. When other essential properties show that the silk fibers of PTRI hybrids perform good in processing, strength, handle, ability to withstand stress and strain in weaving, and physically even, neat, and clean. Next slide, please. Next, please. Therefore, its spread for commercialization was inevitable. The hybrids became the front lines. Next, please. Where pure hand-woven fabrics have become PTR's forefront in support of the development of Philippine silk products. Last slide, please. The commercial distribution and use of PTRI developed hybrids by 100 cocoon producers in Mindanao, next please, have validated the productivity and quality silk properties of the new hybrids, essential to strengthening and furtherance of the Philippine silk industry. Next please. From these 4 productive silkworm hybrid lines, next please, spurred the incessant pursuit of PTRI R&D in developing more and suitable hybrids with a tangible goal, next please, of satisfying at least 10% more every year of the Philippine silk requirement. Thank you very much for listening.
Thank you very much, Ms. Lopez. Okay, now let's move to the last finalist of this cluster, Joanna Manuel, also from the PTRI DOST.
Good morning. Thank you for having me here. And in behalf of my co-author, Dr. Julius Liano, I am pleased to present our paper entitled High Molecular Weight Carrier Stabilized Spray Drying of A2 Stalisay Leaf Extract as Natural Dye for Textile. Next, please. Spray drying. Next, please. Spray drying is a technique used to increase shelf life of the product by converting liquid sample into powdered form. Powders have many benefits compared to its liquid counterparts, such as reduced volume, weight, and packaging. Literatures have shown that this technique can also be applied to natural dyes. In fact, PTRI have already adopted spray drying as method of converting the crude extract into powder dye. However, due to its hygroscopic and sticky features, powder yield is very limited as most of the powder adhere to the dryer wall, and as a result, they are sold at uncompetitive price points. Next, please. To address this dilemma, the following areas may be explored. First is to determine the optimized drying parameter. Second is to improve the shelf life of the powder. And lastly is to enhance the yield of the powder. Next, please. In the food industry, carrier agents are added in the drying operation to improve the property and stability of the powder. Although this method has well been established and practiced in food production, this practice has yet to be applied with natural dye. Next, please. In this study, the researcher primarily aims to identify which carrier agents are suitable for the spray drying of Talisay leaf extract. The carrier used in this study are maltodextrin, tapioca starch, carrageenan, and cream of tartar. Next, please. Next, please. Next. To evaluate the effectiveness of the carrier agents, the following parameters were observed: color fastness, moisture content, water activity, decomposition, and glass transition temperature. Next slide. Next, until the end of the slide, please. Results show that the powder yield is for pure talisay leaf is 27.5 gram, while the yield for pure carriers alone are as follows. However, after the drying process, it was noted that the yield for maltodextrin is 37.6 gram. Thus, based on this, addition of carrier agent gives a significant increase in the yield of the powder, wherein the highest yield was observed in 3% maltodextrin. Meanwhile, reconstituted powder as applied to cotton jersey resembles the same shade to the crude extract, which is the desired result in dyeing process. Next slide, please. Next slide. It was also noted that the powder with tapioca starch has the lowest alteration in moisture content after 30 days compared to pure powder. Meanwhile, the changes in water activity of the powder are inconcurrent with the observation in the moisture content. With this, it can be concluded that the carrier agent significantly lowers the water absorption capacity of the powder dye. Carrier agents also improve the thermal properties of the powdered sample by increasing the glass transition and the composition temperature. Thus, based on this finding, it was noted that the use of carrier agents improved the powder yield by 68%, the thermal properties by 38%, and the shelf life by 12%. Furthermore, among the carrier agents, 0.5 cream of tartar and 0.5 tapioca starch are the effective carrier concentrate for the talisay leaves powder. Thank you. That's all.
Thank you very much, Ms. Manuel. Now, let's move to the last cluster, disease, medicine, and pharmaceutical science. Our first finalist for this cluster is Mr. Kevin Christian Olarte from the National Institute of Molecular Biology and Biotechnology of UP Diliman.
So, hello, good morning, everyone. I am Kevin Olarte, and today I will be presenting our study on the role of cytochrome B561 in neuroendocrine prostate cancer. So, early stages of prostate cancer are usually treated using androgen deprivation therapy. However, after a few years of treatment, cancer cells often develop resistance to therapy, and the disease eventually progresses into a more aggressive and lethal form known as castration-resistant prostate cancer. Gene expression studies have tried to identify differentially regulated genes between the different stages of prostate cancer to call out key genes which may drive resistance and disease progression, and one of the genes which is found to be upregulated in the latter stages of the disease is cytochrome B561 or CYB561 gene. This gene codes for an electron transfer protein found to be enriched in neuroendocrine secretory vesicles, where it mainly functions in neuropeptide synthesis and activation. However, no functional studies have been done to determine the direct role of CYB561 in prostate cancer and castration resistance. So our study sought to demonstrate the possible roles of CYB561 in the lethal neuroendocrine type of prostate cancer. Next slide, please. Essentially, so what we did was to knock down CYB561 in PC3 cells, a cell model representative of the neuroendocrine type of prostate cancer, and looked at its effects in three key areas, namely in the neuroendocrine phenotype and function of these cells, in iron metabolism, and in the overall cell behavior of these cells. So what we found was that knocking down CYB561 decreased the expression of neuroendocrine markers such as synaptophysin and neuron-specific enolase, and decreased the presence of growth or survival-promoting metabolites in the conditioned media from these cells, suggesting that these cells are somewhat becoming less neuroendocrine in nature. Moving on to iron metabolism, ferrous iron is known to be the active form of iron and is often used by cancer cells as important cofactor for different essential metabolic processes. What we found was that knocking down CYB561 also affected ferrous iron concentration by acting as a ferroreductase, since knocking down CYB561 decreased intracellular ferrous iron concentration of PC3 cells, as evident in our direct measurements, and as indicated by an upregulated transferrin receptor mRNA and downregulated ferritin mRNA. Next slide, please. Finally, PC3 cells are known to be very aggressive and proliferative in vitro cultures. However, through different cell assays, we found that knocking down CYB561 decreased the proliferation, survival, and migration of these cells in vitro. So taken together, this is the first study to demonstrate that CYB561 plays a vital role in maintaining the aggressive behavior of neuroendocrine prostate cancer cells through its neuroendocrine and ferroreductase activities. Thank you.
Thank you very much, Mr. Olarte. Now let's move on. The next presenter is Mr. Rock Christian Thomas of the Department of Electrical Engineering, UP Los Baños.
Good morning, everyone. On behalf of the members of the team, I am Rock Tomas, here to present our study titled Discrimination of Malignant from Benign Thyroid Lesions through Neural Networks using FTIR signals obtained from tissues. In the Philippines, thyroid cancer remains one of the leading forms of cancer. Some types of thyroid cancers are difficult to diagnose. Some are overdiagnosed and therefore undergo overtreatment. As a result, these cases can reoccur after several years. The current gold standard in cancer diagnosis, the microscopic examinations of H&E stain biopsies, is prone to bias since greatly relies on visual examination. Hence, there is a need to develop more sensitive and specific methods for diagnosing cancer. Here, FTIR spectroscopy of thyroid tumors were performed and artificial intelligence models via neural networks were designed to discriminate the spectral data. Next slide, please. Next slide, please. Pre-methodology implementation, tissue sections were subjected to ATR FTIR analysis, and two pathologists validated their histopathological state via their corresponding H&E sample. After such, the resulting spectral data were analyzed by 5 neural networks. The general design is shown in number 1, and the preparation of data is shown in number 2, where all data were grouped accordingly to produce 10 different data sets, which are unique from one another. The preparation is then proceeded with the training process, which is shown in number 3. Lastly, LDA and PCA-HC were used as classical benchmarks for comparison. Each model was evaluated using a stratified 10-fold cross-validation procedure to avoid overfitting and to avoid a bias. And the performance metrics, accuracy, area under the curve, positive predictive value, negative predictive value, specificity, and recall were averaged for the performance of each model. Next, please. Among the neural network models, the neural network model, here labeled as RNN-based model, performed the best, having a near-perfect AUC, PPV, and S values, while an accuracy of 99.41%, an NPV of 99%, and a recall of 98.75%. All neural networks, nonetheless, had performed excellently as classifiers. In conclusion, neural network-based model analysis of FTIR spectral data is potentially an objective and specific method for diagnosing thyroid cancer. Overall, the study shows a promising incorporation of artificial intelligence, not in supporting thyroid cancer, but potentially for all cancers as well. Thank you.
Thank you very much, Mr. Tomas. Next, we have Charina Gail Baloy of the Graduate School, University of Santo Tomas.
Good morning, everyone. May I ask the host to present all the animations for this slide, please? Okay, so my study is about in vitro anti-beta secretase, anticholine esterase, and free radical scavenging activities of Clitoria ternatea lymphabaceae leaf extract. Alzheimer's disease is the most common neurodegenerative disease, mostly associated with cognitive impairment and behavioral disability. Currently available drugs for it is based on inhibition of acetylcholine esterase and NMDA receptor. However, these drugs cannot be considered as radical treatments because they only offer symptomatic treatment. So because of this, AD research is turning towards the use of natural compounds in an effort to discover new therapeutic agents for the disease. So specifically, Clitoria ternatea, also known as butterfly pea, has long been used in Ayurvedic medicine for CNS disorders. Although this plant have shown to improve cognition and memory, the exact mechanism behind its effect remains unclear. So my study investigated the anti-Alzheimer's effects of this plant. Next, please. Until the end of the slide, please. Next. Okay, so to do that, the powdered leaves extract or the powdered leaf sample of this plant is sequentially extracted by solvents with increasing polarity, starting from hexane, ethyl acetate to methanol, and then all the extract are subjected to in vitro assays for the determination of their beta secretase, acetylcholine esterase, nitric oxide, and DPPH radical scavenging inhibitory activities. And then the most active extract was then subjected to phytochemical analysis by thin layer chromatography, total phenolic content determination, total flavonoid content determination, and liquid chromatography mass spectroscopy for the determination of the active metabolite. Next, please, until the end of the slide. Thank you. Okay, so based on the results, the methanol extract exhibited the highest inhibitor activities on all the enzymes and free radicals. So based on its composition, the methanol extract also showed the highest phenolic and flavonoid content, which can account for its markedly higher in vitro activities as compared with the other extract. TLC analysis showed the presence of phenols, flavonoids, and sugars, and this was confirmed by the results of LCQ of MS analysis, showing 4-caempheryl glycosides as the main component of the extract. So in conclusion, my study confirmed or supported the traditional use of the plant against Alzheimer's, highlighting its effect on beta secretase, acetylcholine esterase, and free radicals. This is also the first report of the 2 out of the 4-caempheryl glycosides that was detected in the extract. So that's all. Thank you all for listening.
Thank you, Ms. Baloy. Okay, next we have Mr. Omar Villalobos of the Department of Pharmacy, UST.
Kindly show all the animations for these slides. Thank you. Good morning, everyone. I'm Omar Villalobos from the University of Santo Tomas, and I will be presenting my research entitled Euglycemic and Wound Healing Activities of Vitia antidysenterica extracts. So first, diabetes is a chronic and progressive endocrine disorder in which the amount of glucose in the blood is not properly used or distributed in the body. According to International Diabetes Federation, there are around 463 million people afflicted with diabetes and will continue to rise to 700 million by the year 2045. One of the management to decrease the absorption of glucose in the body is to inhibit the carbohydrate hydrolyzing enzyme, alpha amylase and alpha glucosidase. Next slide, please. For the methods, to determine the euglycemic and wound healing activities of Vitia antidysenterica, the following methods were performed. First, to screen out the extract which inhibits the alpha amylase and alpha glucosidase. In vitro inhibition assay was done. Next, in vivo assays were performed to determine the effect of the active extract to different diabetic and wound-inflicted zebrafish model. Finally, the phytoconstituents of the active extract were identified through LCMS. Next, please. Results of my study. First, for the in vitro inhibition assay. First, the active extract was identified as the Vitia antidysenterica bark dichloromethane extract with an IC50 value of 0.1366 mg per ml against alpha amylase and 0.2933 mg per ml against alpha glucosidase. So inhibition of this enzyme would result in the decrease of postprandial glucose absorption. Next, for the in vivo anti-hyperglycemic assay, 3 zebrafish models were used: leptin knockout, zebrafish, an alloxan-induced model, and STZ-induced model. So the 3 models were used to evaluate the in vivo anti-hyperglycemic assay. So 2 concentration of the WAB-DCM or the active extract was able to decrease the postprandial glucose level of the zebrafish model. For the in vivo wound healing activity, it resulted to the increase of wound closure of zebrafish, laser-inflicted wound model, which was observed from day 10 up to day 30. Finally, the phytoconstituents identified through LCMS QTOP were two unknown alkaloid and 2 polyunsaturated fatty acids. Based on the related literature, these phytoconstituents can help in the management of diabetes. That would be all. Thank you.
Thank you, Mr. Villalobos. Last but not the least, we have Mr. Romy Angelo Azur of the NIMBB UP Diliman.
Good morning, everyone. Androgens mediate the growth and development of the adult prostate by binding to their cognate receptor, the AR. Upon activation, AR translocates to the nucleus where it associates with DNA elements, and there it recruits co-regulators to facilitate expression of target genes. Next, please. Aberrant signaling in this axis results to uncontrolled proliferation and ultimately to prostate cancer. Next, please. Due to the prominence of AR signaling in carcinogenesis, androgen deprivation therapy is used as a primary care regimen for managing prostate cancer. Next, please. However, most patients develop resistance and the cancer ultimately recurs towards a therapy-resistant state termed castration-resistant prostate cancer or CRPC. Surprisingly, AR signaling has been detected in CRPC and has been postulated to mediate its development. Next, please. Current drugs are ineffective against CRPC, highlighting the need to develop innovative treatments to delay therapy resistance. To address this need, we adopted a luminescence-based bioassay that can identify novel AR antagonists. Next, please. The screening pipeline was first validated by performing a control plate where we saw high dynamic range to discriminate putative AR antagonists. Next, please. Next. We then proceeded with the screen. Samples that were able to inhibit AR activity by at least 75% without decreasing cell viability by more than 50% were considered as candidate hits. From the 152 samples we tested, only one, extract A32, was able to satisfy this criteria. Next, please. Next. The activity of A32 was then validated through a dose-response assay where we observed prominent dose-dependent inhibition of AR activity. Next. Supporting its status as a bona fide candidate hit. Next, please. To determine its bioactive component, A32 was subfractionated and subjected to the same screening assay. We observed that fractions F6, F7, and F8 significantly reduced AR activity, implying that the three subfractions may contain the bioactive components of A32. Next, please. Next. The fractions were then subjected, The fractions were then subjected to tandem MS analysis and OPLSDA to identify the bioactive metabolites. Our analysis was able to cluster the active subfractions from the rest, and looking closer, 8 compounds were predicted to be major contributors to A32's activity. To provide proof of concept on their mechanism of action, molecular docking simulations with the ligand binding domain of AR was performed. Simulations predicted candidate compounds to bind to a pocket adjacent to the co-activator binding platform AF2. Next, please. This suggests that the anti-AR activity of A32 may be facilitated by the bioactive metabolites through inhibition of AF2 formation and suppression of co-activator recruitment. These ultimately impedes AR-mediated transcription. Thank you.
Thank you very much, Mr. Azur. Okay, so that's it. Congratulations once again to all our finalists. You all did a good job. And of course, we would like to thank all the members of the Board of Judges for working hard for this session. Now, what are the prizes at stake for the winners? Again, as I mentioned earlier, we will be selecting, the judges will be selecting the top 3 for each cluster. For the third place, he or she will receive 1,000 pesos. For the second place, 3,000 pesos. And for the first place, 6,000 pesos. So I will be communicating with the winners after we announce them on Friday on how you can actually get your prizes. Okay, so again, announcements of winners will be done during the closing ceremonies of this 40th APAMS. And that will be on Friday, August 14 at 8 a.m. So I invite you all to please be present in that session, especially our finalists. You may register, of course, to the link that I have provided you through email, or you can check that once again in our paase.org website. Okay, so thank you very much once again. Okay, maybe we can have a few words from our president, Dr. Giselle Concepcion.
Thank you, Dr. Rivera Windle, for organizing this rapid fire competition, to our judges and to all our participants. And we're happy to see a level of interest, enthusiasm, and competence in your research. The quality of research in the Philippines is improving and it's promising. And it's nice to see participants from other institutions like the DOST-PTRI, University of San Agustin, and of course we have the UP constituent universities, but also from the University of Santo Tomas and Ateneo de Manila University. Thank you all, and please join us on Friday so we can know who the winners are and celebrate with them. Thank you.
Thank you, everyone.